Thank you. Thank you very much. Thank you very much, Dr. Hill, Russell. Thank you for giving me this opportunity to speak to the audience today. Welcome, everybody. This is an unusual format where we will have 3 speakers today. And as Dr. Hill said, we'll be joined by Victoria Laye, who is, I think, our 9th or 10th graduate student from the lab. And also Priya DasSarma, who's been a longtime colleague of mine. We've worked together for over 25 years. And the title of today's talk is Evolution and Survival of Ancient Microbes. And these ancient microbes that we study are called halophiles, and they can exist very deep underground, above the atmosphere. And recently, there's been a lot of interest in whether they might even survive on Mars. And you can see on the panel on the right is a photograph of Mars, a NASA photograph, and up to the upper right-hand corner is not a moon, but it's actually a snapshot that Priya took last week as Mars came in opposition to Earth. It's actually very close, the closest it gets to Earth, and so one can see it with the naked eye. So as I mentioned, the organisms that we work on are called halophilic archaea, or haloarchaea for short. Halo is from the Latin salt and the sea, and so they are sea or salt-loving archaea. And in fact, actually, they cannot exist in the sea because the salt concentration is too low for them there. And these organisms are members of the 3rd domain of life, the archaea. And on the left side, you can see a cartoon of an evolutionary tree that's lined up next to a timeline of the history of the Earth. The Earth is about 4.5 billion years old. And evolutionary biologists think that life originated very early in the first billion years, or at least in the 2nd billion years. And all life, all extant life that we have on the planet evolved from an original common ancestor, which sometimes is called LUCA for last universal common ancestor. And this organism was already cellular, and it had some features that are universal. It had a charge separation mechanism to make ATP, called chemiosmotic coupling, for energy for the cell. And it had a genetic code that essentially has survived to this day. So all organisms that we find on Earth, whether they're archaea, bacteria, or eukarya, have essentially the standard genetic code. What exactly happened to give rise to these 3 lineages is not entirely clear, but what we do know is that the haloarchaea, like the other archaea, likely evolved very, very early. And so you might think of them as relics of a very early time on Earth. And in the center, you can see the orange color inside of a crystal of halite or sodium chloride. And they can indeed survive embedded in halite for an extended period of time. And so these organisms, we've studied them for about 40 years now. I started working on them at MIT in 1980. And they have provided really a great way for us to have insight into a number of different fundamental aspects of life, including its unity on Earth, as well as its diversity and its limits, which is a major topic of today's presentation. And toward the end, we'll talk about its distribution and also origin. So these organisms grow in very high salinity. And on the left, you can see a video of a culture of haloarchaea that has just been allowed to evaporate. And you can see how quickly that the salts form, the salt crystals form. On the right-hand side, the same process would occur with seawater, which is lower salinity, but ultimately the salts would become concentrated, and initially there would be a precipitation of calcium sulfate or gypsum, followed by precipitation of sodium chloride or halite, and then ultimately potash. When the concentration of salts gets to the point of sodium chloride precipitation, one generally considers that the limit to life. There is really very few life forms, if any, that can survive beyond that point. And this is shown in the next slide. So this is showing the growth rate of different organisms versus the sodium chloride concentration. And on the left-hand side, you can see the salinity of seawater is around 0.6 molar. And a typical marine organism like the cyanobacterium PR6 will have an optimum that's right around the seawater concentration, and it won't be able to grow above about 2 molar sodium chloride. On the right-hand side, in the dark blue, you can see the haloarchaea. These are the most salt-loving organisms known. They can grow at about above 2.5 molar, so about 4 times the salinity of the seawater. And in fact, they will lyse or break open below that concentration. Their optimum is within 4 to 5 molar sodium chloride. And they can grow pretty well even at saturation, which is about 5.3 molar. So this is about 25% salt by weight. So where did I get introduced to these unusual organisms? Well, as Dr. Hill mentioned, we were introduced to this at MIT, and I had the good fortune of having 2 advisors, not one. Dr. Khurana, who's a Nobel laureate in 1968 for deciphering the genetic code. He shared that award with 2 other scientists, and also Professor Raj Bhandari. When I joined their laboratory, it was in 1980, and they had moved on from working on the genetic code to other universal features. And in particular, Dr. Khurana was interested in how ions move across the membrane, because that was a very fundamental aspect of this chemiosmotic coupling that cells use to make energy. And he was studying a very interesting membrane called the purple membrane, which is produced by the haloarchaea. And you can see the culture is really purple on the right. And he handed me a culture of this organism, and also showed how the purple membrane can be easily purified. So this is a very simple and ancient membrane, the purple membrane, and it's formed by an addition of a chromophore called retinal, which is very similar to vitamin A, to an opsin called bacteria opsin. So this one-to-one adduct forms a protein called bacterodopsin, which behaves as a transmembrane light-driven ion pump. So it pumps hydrogen ions across the membrane, as shown in the center. This creates what's called a proton motive force. Hydrogen ion is basically just a proton. So that pumping of the proton across the membrane that you can see on the right can result in a potential energy that can be used by the cell to produce ATP, which is its energy currency. And this was actually an experiment that was done by 2 other scientists, Racker and Stokenius, which showed that chemiosmotic coupling actually occurs in cells, because this was a controversial idea before. So we've been focusing recently on the fact that the pigment is purple. Why is it purple? Well, it's purple because it absorbs green light and it reflects red and blue light. So this is a program from NHK called Cosmic Front, and they came and they talked to us about our research and produced, this is a part of this program that shows why the purple membrane is purple. So you can see here is a demo. You take out the green and you combine blue and red, you get the color purple. So it's as simple as that. So spectroscopically, the purple membrane has the spectrum shown in the panel on the left, and you can see that there's an absorption maximum around 568 nanometers, and it doesn't really absorb at the two ends of the spectrum around 400 or 700, that's the visible range. And so it's absorbing green light, it's reflecting or transmitting blue and red light, and so therefore it's purple. One of the things that we noticed was that the spectrum of purple membrane is complementary to another very common pigment, the chlorophylls, which are found in plants, of course. And these, everybody knows, are green. And one of the questions that people pose is, why are plants green? And we came up with a hypothesis that would explain that, and we call that the purple earth hypothesis. In this case, what we think is that there was a co-evolution of these two pigments, the retinal purple pigment and the green chlorophyll pigment. If the purple pigment occurred first and absorbed the sunlight in the green-yellow range, then it might have left only the blue and red parts of the spectrum for the evolution of green chlorophyll. And this idea has sort of taken hold and has been explained in programs such as on the PBS EONS and the CNN Color Scope, and also in the NHK program. So the other feature, which I'm not going to talk about in detail today, are the gas vesicle nanoparticles. And you can see on the right that the cells can float as a result of these buoyant particles that are present in the cell. And we've been using these for biotechnological development. And we call them GVNPs for gas vesicle nanoparticles, and they have very interesting and valuable properties. They're very easily purified by flotation. They're completely non-toxic. They're incredibly stable, so one doesn't need any cold storage to keep them. And they're genetically engineerable. And so you can see on the right, the lemon-shaped particle has a surface where, which serves as a platform for display of other proteins, foreign proteins. And we've been using this for therapeutic protein delivery and for antigen delivery for vaccines. And I just did a webinar last month that was sponsored by the scientist. And if you want to know more about this, this webinar was recorded and is available for viewing. So I want to get more into the halophilic archaea or the haloarchaea. So these organisms are very common. They are found wherever there's salt, and there's salt all over this world. And we've gone to a lot of different environments to study them. Some of these environments are very old, such as these Permian deposits that are 250 million years old, such as in Carlsbad, New Mexico, or in South America. Some of them are not that old. They're tens of thousands of years old, such as in the Great Salt Lake, Salar de Uyuni in South America, and Deep Lake in Antarctica. And then others are very recent because people mine salt from the sea, such as in San Francisco Bay in the west and Inagua, Bahamas in the east. So I want to just show you a few of these environments because they are so beautiful and striking, and the organisms are pigmented and produce visible signatures. So this is an aerial view of San Francisco Bay. Those of you who have flown into San Francisco may have seen these ponds because the flight pattern takes you right over these. They're so beautiful that there's a whole series of photographs, some of them taken by Robert Campbell Photography, which are actually sold commercially. And these have become popular for the covers of magazines, such as an article that we published in the American Scientist, The Salty Side of Life. And you can see that the Robert Campbell photograph is there. We've also had artists who've been very interested in this organism, and one very well-known artist, Michelle Hagewood, who was at the Walters Art Museum in Baltimore, and then moved on to the Guggenheim and then the Metropolitan Museum of Art in New York, came to our lab as an artist in residence, and she produced some very beautiful paintings, as you can see, 2 of them in front of you. So when the Journal of Bacteriology asked us for a cover photo for their cover, we had a really hard time deciding which one. So ultimately, we decided the one on the right was the one that we chose. So this was the first real art piece that appeared on the cover of this scientific journal. Another very spectacular environment for haloarchaea is the Great Salt Lake. It's the largest salt lake in North America, and you can see the red brine. It also served as a canvas for art. In fact, Robert Smithson is famous for his spiral jetty, as you can see on the right side. This is an example of eco art. It's essentially using the earth as a canvas. And in fact, even astronauts in space found it so beautiful to photograph it. This is the Great Salt Lake, and the north end on the right-hand side is red from growth of halobacteria or haloarchaea. And this one we published on the cover of the American Biology Teacher. And the reason for that was that we have a number of resources for using these haloarchaea for teaching, because archaea in general and haloarchaea in particular are not pathogenic. They are ideal for teaching children. And so you can see here are some of the kits that we have. So if you're interested in doing some biology at home, this is something that you can do. And we also have a website which was which we put up with the help of our son, who's a computer scientist. And you can see here that there's information on the various kits, as well as Mulgent, which is a video teaching resource for students. Okay, recently we've gone to South America through our collaborators to the Bolivian Altiplano, which is a very high desert, which contains a very, very large salt flat. It's about the size of New Jersey, I think. And at Earth level, it looks like this. It's a very flat white area, and there's no life on the surface, but just beneath the surface, as you can see from these blocks, there are microorganisms that grow. And then finally, I want to take you to Carlsbad, to the Permian salt deposits. And this is thanks to Dennis Powers, who took us on this tour last year, and also Brian Dozier, who provided the video here. I can show you, actually take you down into this mine. So, whoops. Okay. Okay, here we go. So this Permian salt deposit is about 0.65 kilometers below the ground, about 2,100 feet below the ground. That's about 1.5 times the height of the Empire State Building. And that's where there's a very thick layer of salt that was deposited about 250 million years ago, before the time of the dinosaurs. And they're actually using this very geologically stable region for storing nuclear waste from power plants, from nuclear power plants. But for us, it's a great place to get salt with very old microorganisms. So you can see as you step off the elevator, they have these tunnels, and they use these for storage of nuclear material. And so this is a new tunnel that's been dug, and you can see the walls are all made of salt. So you can see what the walls look like here. These are halide crystals, and they've been deposited over time. And WIP was kind enough to provide us with samples for isolation of organisms. So we've been doing this, as I said, for close to 40 years, and we have a lot of different organisms that we have in our collection. And we have a database, which also was, again, put together by our son in the UMBC, halo.umbc.edu. And we've done a lot of studies on the genomics and molecular biology of these salt-loving haloarchaea. We've isolation and characterization, genome sequences. We did the very first genome sequence along with a consortium of researchers back in 2000, which appeared on the cover of the Proceedings of the National Academy of Sciences. We've also done a lot of transcriptomics and genetic mutations, and as I mentioned, biotechnological applications. But today we're going to focus on the survival studies, which are astrobiological in nature. And these organisms tolerate not just salty, dry conditions, but also several other conditions that Victoria and Priya are going to tell you about. And there are two model organisms that we've been using for these studies. One is Halobacterium species NRC1 from San Francisco Bay, and the other is Halorubrum lacus profundi, or HLA, from Antarctica. And the one, NRC1, is a mesophile. It grows essentially in warmer temperatures, a little bit above body temperature optimally, and Halorubrum lacus profundi can survive and grow even in sub-zero temperatures. So take it away, Victoria.
Okay. So haloarchaea are able to grow using a number of different types of energy. They can use light through photophosphorylation, which you can see in that blue box. So that's the process used by bacteriorhodopsin in the cell membrane to transform light energy into usable energy by pumping out hydrogen ions, creating an imbalance, like Dr. DasSarma said earlier. They can also use fermentation, which is the green box, by breaking down amino acids into their component parts, releasing energy that can then be used to form ATP, which stores the energy for use in other cellular processes. And finally, they can use either aerobic or anaerobic respiration, which is the red box. Aerobic respiration uses oxygen to pump hydrogen out of the cell, much like photophosphorylation, whereas anaerobic respiration occurs when there is no oxygen available, so a different electron acceptor must be used instead. This occurs in areas deep underground, like in salt mines, where there is limited light and oxygen, but high salinity, so a reasonable place to find haloarchaea. We found that perchlorate can be used by some organisms in situations like this, which is interesting because magnesium perchlorate is suspected to be present on Mars, and ammonium perchlorate is used in rocket fuel. It reacts with itself to break apart into its component ions and release a great deal of energy. So you could say that some haloarchaea can survive on rocket fuel. Next slide. We grew both Halobacterium NRC1 and Halorubrum lacus profundi, or HLA, under anaerobic conditions with and without added sodium perchlorate. So as you can see in the graph on the left, we found that after 8 days, NRC1 had not grown at all under either conditions, which is the hatched bars, while HLA was able to grow better with the added sodium perchlorate, the solid blue bar, rather than without it, which is the solid red bar. So that suggests that HLA is able to use perchlorate when growing anaerobically. We also measured aerobic growth, which is on the right, with added magnesium chloride, sodium perchlorate, and magnesium perchlorate to test the effects of magnesium and perchlorate on the haloarchaea, as they're generally thought to be limiting to life, and we were specifically looking to see how much we could put in there to cause a complete stoppage of growth. So what we found was that perchlorate had the largest reduction in growth, so that's the green line, followed by sodium perchlorate, which is the blue line, and then magnesium chloride with the smallest reduction in red. So that suggests that the negative effects on the haloarchaea are mainly from the perchlorates. So by testing both aerobic and anaerobic growth, we were able to see how the growth could both be helped with a small amount and hindered if you add enough, depending on what other limiting factors are also imposed on the organisms. Next slide. So we also used our model halophilic enzyme beta-galactosidase from HLA to test the effects of magnesium and perchlorate on activity. You can see a model of this enzyme rotating on the right. So beta-galactosidase can be used in processing lactose-free milk as it's able to break down lactose. So beta-galactosidase works well for these kinds of tests because it has a simple visual assay where the substrate, ONPG, is broken down into ONP, resulting in a color change that allows us to see how well the protein is functioning. And we can quantify this using steady state kinetics, where you add enough substrate to be able to assume a steady state where the enzyme always has substrate bound, and we can extrapolate from that how quickly the enzyme is converting that substrate into the product, giving us an idea of how well it's functioning. Next slide. So we calculated the inhibition constant for each salt. So that tells us how much of the salt is needed to bring the protein down to half its optimal function. So the graphs also give us an idea of how that inhibition is happening. When we tested the effect of magnesium and perchlorate on the function of the enzyme, we determined that magnesium perchlorate, which is in the red box, again had the largest deleterious effect with a Ki, or an inhibition constant, of 0.04 molar. That mainly comes from the magnesium, as magnesium chloride had a Ki of 0.3 molar in the blue box, and sodium perchlorate in the green box had a Ki of 2 molar. So that means it takes significantly more sodium perchlorate to see the same amount of inhibition that you see with magnesium chloride or magnesium perchlorate. This shows that depending on where the salt ions are found can change how the cells are affected by them. Since the main effect seems to be coming from magnesium in this case, while in the growth experiments it was perchlorate, we can hypothesize that magnesium is not able to pass through the cell membrane, or it may have a larger effect on cell growth if it was able to actually pass into the cell. Next slide. So the enzymes from haloarchaea are unique in that they are highly acidic or negatively charged. This helps them to remain in solution even when in the presence of a lot of salt, like inside a haloarchaeal cell. As you can see here, this high red peak indicates that the majority of proteins from haloarchaea are around 4.2, while non-halophiles have an acidic peak and a basic peak, with the average being much more neutral. The acidic peak for non-halophiles is also not as acidic as the halophile peak. Next slide. So you can see the highly acidic surface in the HLA, that's the beta-galactosidase on the top, represented by the red highlights. So those indicate acidic residues compared to a homologous beta-galactosidase from a non-halophilic organism, where you can see there's both blue, which would be basic residues, and red, which are again acidic residues, in an equal measure, making a much more neutral surface to the protein compared to the halophilic protein, where it's very acidic. So haloarchaea are very well adapted to living under conditions like those underground, where there may be low light, low oxygen, or high salt, thought to be life-limiting, both in a general sense and when looking specifically at their proteins. So there's less available energy and the potential for dehydration, but haloarchaeal organisms have developed strategies to combat both. So they're able to survive all that and so much more, as Priya will be able to tell you.
Yeah, so my favorite topic, haloarchaea. Haloarchaea are really versatile. They can handle all kinds of stressors and are adapted to handle extreme environments using tools like the acidic proteins Victoria just mentioned, and we discovered when we sequenced the genome of the first haloarchaea on NRC1. They can even survive in brine inclusions in salt crystals like the ones you see here. In fact, the Washington Post even called them superbugs due to their amazingly resilient qualities, and superbugs in a good way. Next one, please. Haloarchaea are found everywhere in the world, underground, on land, in brine ponds and lakes, but how have they gotten there? There are some studies showing that halophiles can be transported in dust storms across the oceans high up in the air, and that over a trillion, and for those of you, I had to look it up, it's 10 to the 21st cells are lifted annually into the atmosphere. To give a bit of perspective, once you leave the Earth's surface, there are multiple layers of the atmosphere. The first three, someone's got a microphone on. Could you turn it off, please? Thank you. So to give you a bit of perspective, so once you've left the Earth's surface, there are these multiple layers of the atmosphere, with the first three being the troposphere, where most of our weather occurs. That's what's around you, which rises up to about 12 kilometers. And the temperature actually goes down as you go higher up, as altitude increases. And there are many microbes floating around here. You know, some of the ones that we don't want, some of the ones we do want. The next layer, the stratosphere, is less well studied. And this is where jet planes fly, where we find the ozone layer, which some of you who are old enough to remember the ozone hole that went away and then now apparently is returning. The ozone layer is about 32 kilometers above sea level, and that's what protects us from the most harmful of UV radiation. Above it, and still in the stratosphere, is where the UVC radiation is and where you can find weather balloons. At about 50 kilometers, you reach the mesosphere, which extends to 120 kilometers, and you find temperatures here as low as minus 85 degrees Celsius. This is the layer where meteors burn up and the very highest clouds are found. And for those of you who think Fahrenheit, which I cannot, being a scientist, minus 20 is what your regular freezer would be. So minus 85 is a lot colder. But back to our supermicrobes. We've seen what these microbes can do underground and using unusual chemicals. Let's see how far out we can push them to see where else they can survive. If you can go back just a moment, please. Can you go back to the previous slide, please? Thank you. So this is a view from a plane that our son took flying over San Francisco Bay, and you can see the Halobacterium growing, floating due to the gas vesicles that we had originally talked about, floating down there in the right-hand corner. Nice, bright reddish. So the colors that you saw in the previous slides are real. I saw them up, you know, we see them at the plane. Next one, please. Now, imagine what it's like outside the plane and higher up in the stratosphere, like where a weather balloon can travel. It's cold. There are high levels of UV radiation, including above the protective ozone layer, where in addition to UVA and B from the sun, there is damaging UVC. It's not very hospitable. And add to that the fact that there's very low pressure, little moisture, and there are virtually no nutrients. Could our microbes survive these conditions? Next, please. Well, along with the Earth to Sky Calculus High School team in California, we sent our haloarchaea up in a weather balloon to see how they would fare. They were live in media, which with a high salt content remains liquid down to minus 20 degrees Celsius. So this is, you know, so they went up to 34 kilometers above sea level, almost twice as high as the plane ride we just took. They encountered temperatures as low as minus 65 degrees Celsius, high UVC doses, and pressures as low as 0.6% that found on Earth's surface. And the big question is, did they survive? Yes, they did survive really well. We compared the cultures that we stored in the lab with those flown to California and back to Maryland. If you can see on the slide, I have it divided into the Halobacterium species NRC1, which I call NRC1, and Halorubrum lacus profundi, the Antarctic strain, which I call HLA on the right-hand side, the three sets. So we compared the cultures stored in the lab with those flown to California and back to Maryland, with those that flew to California, went to the stratosphere, and then were returned to our lab, and those I named balloon. As you can see, there was little effect, though the Antarctic strain, HLA, survived the trip to the stratosphere a bit better than the San Francisco Bay strain, NRC1, and we wanted to see why this was. Next, please. So we decided to test several of the stressors encountered in the stratosphere in our laboratory. Now, remember, I mentioned that the stratosphere is where one encounters all this radiation, and above the ozone layer where this balloon went with the samples, this included extremely harmful UVC, which is used in germicidal lamps. NRC1 did better than HLA in surviving the UVC doses we exposed them to, so that wasn't the reason why HLA survived better. And as it turns out, our studies have shown that NRC1 is really resistant to UV. Since we know it floats up to the surface of its briny environment, it should not surprise us that it has developed many strategies to survive the harmful DNA damaging effects, including using the antioxidant carotenoid pigments, bacterial rubrins, and carotene, and DNA repair proteins, such as single-stranded DNA binding and recombination and repair proteins. On the other hand, HLA, which lives in the sediment, probably doesn't encounter much of UV, so this may explain why UVC is more deadly for it. Next, please. What about the effect of the drop in pressure to less than 1% of that found on Earth's surface? We pushed our microbes, we tested them by putting the cultures under vacuum pressure to see how they would do. We know that the microbes can grow using sunlight in a simpler than photosynthesis way, as you heard, phototrophically, using their purple membranes, and even without any oxygen, anaerobically. But could they survive vacuum? Well, yes. HLA survived a little bit better than NRC1. Perhaps this tolerance gave us some advantage in the stratosphere trip. Next, please. Well, and then cold. What about the cold? We froze our cultures and then thawed them out to test. Now, when I say froze, we took them down all the way to minus 80 degrees. So almost as if they'd gone into the mesosphere. And then we thawed them out and then tested them. Our results showed, not surprisingly, that HLA did drastically better. We sequenced its genome and found that it has more genes involved in cold response and forms protective clumps of cells, which you can see down on the left-hand side, the sort of the clump that you're seeing there, the sort of tangly material with the circles-like material. Those are the cells, and they form what's called a biofilm, and they form lumps. And we think that perhaps this gives them some kind of protection, sort of huddled together. And it also produces more flexible proteins to cope with the cold, something we just recently published in PNAS. All this comes in handy since 8 months in the year, their environment is below freezing, and it gets as cold as minus 20 degrees Celsius, again, like in your freezer. While NRC1 lives at a balmy 10 to 15 degrees Celsius all year round. I mean, it lives in San Francisco. So, next slide, please. So one could say that these microbes are sort of superbugs or polyextremophiles or polytolerant, loving or tolerating many extremes. They can handle the stressors in the stratosphere, which, as it turns out, is about as close as we can get to a laboratory model for Martian conditions, only without lab benches. And here you can see them. This is actually a video from them being sent up into the stratosphere. So this is from the weather balloon. I think you're muted. Oops.
Okay, so the last topic we want to discuss is whether haloarchaea might be able to survive on Mars. And as Priya just mentioned, the conditions in the stratosphere are pretty similar to those on Mars. It's very cold. There's a lot of UV, especially UVC. There's also very low levels of oxygen and pressure, and it's incredibly dry and with very low water content. So we had a meeting recently sponsored by NASA to discuss exactly this point. Now, is there the possibility of extant life on Mars? And the conference participants came up with 4 different locations where there may in fact be extant life. And these are in regions that would be sort of refuges for life, because if it's very dry and harsh nature, they would have to find a refuge. So one of the refuges is within ice. There's mid-latitude ice and there's also polar ice. And in fact, there are brine lakes that have been discovered below the polar ice. And even in mid-latitude ice, there's going to be some brine associated with ice veins that would form within the ice. Another location where life might exist is within caves, and these may be lava tubes or insoluble rocks, and there may also be brines associated with those. And there might also be a considerable amount of brine deep subsurface, 1 kilometer or further below the surface. And so under those conditions, it's also possible that there might be some life that survived. But of course, the one that we're most interested in are the salt deposits that are right on the surface and which are highly accessible to exploration using rovers. And because a lot of the salts are deliquescent or hygroscopic, they actually would take up water out of the air, moisture out of the air. And so they might provide microenvironments where organisms could survive, especially halophiles or haloarchaea. So are there halite deposits near the surface? Well, the radar images show that there are maybe 600 sizable deposits on the surface. And those you can see above and below is the Salinity uni. And you can see that if you dig down below the surface, there are pigmented organisms that can be cultured and survive. So if they survive in Salinity uni, is it possible that they also survive on Mars? How similar are they? These are images of a evaporite on Mars on the left and a Salinity uni on in South America on the right. And although the Salinity uni is quite a bit larger, they have their resemblance to one another. So how would we discover life if it existed on Mars? One way would be to look for lipids and pigments which might exist. If it's extant life, then the pigments would be a very telling feature. And molecules like retinal or beta carotene or bacteriorubrins, which produce these red pigments, would be readily detectable through visible spectra, whereas the membrane phospholipids, which would not be pigmented, would be detectable using a spectroscopy called Raman spectroscopy. Now, it's also possible that pigments could be detected remotely. And we've published a paper recently with a collaborator, Eddie Schwitterman, that suggests that we might be able to see something called a green edge, which is the reflective edge from these types of pigments. And these you can actually detect on Earth from space-based telescopes or air-based telescopes. And this principle may also be applicable to planets that are far away, such as the exoplanets. These are planets that are not in our solar system. They're in other solar systems in the galaxy. And in 2006, we published a paper with Neil Reed and others at a space telescope which showed that there are quite a few planets that are within what's called a habitable zone and might, in fact, have life. There were about 150 planets discovered in 2006. And as of last year, I think there are more than 4,000 that have been discovered. So this is a very active area of research. So I want to leave you with the question here from our studies of halophiles. Does life exist elsewhere, or are we all alone here on Earth? And just to point out a few numbers here, our galaxy is quite vast. It's estimated that there are 10 to the 11th or 100 billion stars. And the universe has maybe 100 billion galaxies. And astronomers have predicted that each star likely has one or more planets. And there are more than 4,000 planets that have already been discovered. And if you extrapolate, astronomers think that there are 40 billion Earth-like planets in the universe. So are we alone? Well, for the moment we are, but over time, you answer the question. So finally, I think that our research over the last 40 years has provided some interesting insights into life, its unity, its diversity, its limits, its distribution, and possibly its origin elsewhere in the universe. So this gives you a little flavor of our research. And as I said, there have been a lot of students over several decades who have participated in this. And I don't know that I can read everybody's name here, but Priya and Victoria obviously have contributed a lot. We have Fabiana Martinez and Brian Bergquist, Sean Kennedy, Chin-Fen Yang, Victor Ying, Mindy Capes, John Myung Kim, Nitin Baliga, and then our son Satyajit has been pretty active, although he was not a student in the lab. He was an intern. Ram Karan, James Coker, Jochen Mueller, Ham Shukla. And then I showed you the beautiful paintings of Michelle, and these others are collaborators. Tony Phillips helped us with the Stratosphere experiments. Daniel Guzman in Bolivia. Wolf Pescher here in Baltimore has been a very close collaborator. David Crawley in Massachusetts. Joan Slonczewski and Linda DeVoe. Elizabeth Balser-Kubicek. Eddie Schwitterman. Kelly Toole in Kansas, and recently Betul Kakar. We also have collaborators in New England Biolabs who've been very, very helpful with sequencing genomes, Brian Anton and Sir Richard Roberts. Also Shirley McCready and Ivan Bubriek in England, and at Space Telescope, Neil Reed and Bill Sparks. And New Mexico and Texas, Brian Dozier and Dennis Powers. And we have some very interesting collaborations recently with Vince Volz and Bob Stanley in Temple. And of course, Carolina Biologicals, which will make the haloarchaea available to you. So I think I'll stop there. And if there are any questions, we'd all be happy to answer them. Thank you very much.
Thank you. Thank you to Professor DasSarma and to Priya and Victoria for a fascinating talk. And please go ahead if you have any questions for our speakers. If you type those in the question and answer box, I'll be able to put those forward. But let me start off with one for Professor DasSarma. You showed that map of the distribution of